Okay, okay. All right, so thank you very much, Dr. Andrew Stanfield, for joining the webinar today. We at SRF are very excited to continue the webinar series. Just as a reminder, the goals of the series are to get all of the parents and caregivers closer to the science of SynGAP 1, making everyone aware of the research that's currently being done and opportunities to participate in that research. And finally, empowering patient families in their communications with their current clinicians. As I mentioned, today's speaker is Dr. Andrew Stanfield, and his discussion is titled Cognition, Behaviour and Clinical Trials in SynGAP 1. Dr. Stanfield is a clinical research fellow and honorary consultant psychiatrist at the University of Edinburgh. Following the completion of his training in psychiatry, Dr. Stanfield took up a position as a clinical lecturer at the University of Edinburgh. Within this post, he obtained a Wellcome Trust Clinical Research Training Fellowship to examine the relationship between autism and the schizophrenia spectrum. Following this, he was appointed as a Senior Clinical Research Fellow in the Division of Psychiatry, and he is the co-director of the Patrick Wild Centre. He undertakes clinical work as an honorary consultant psychiatrist for NHS Lothian Learning Disability Services, including sessions for the Regional Autism Spectrum Disorders Consultancy Service. He's also a founding member of the Taylor Ed Foundation, a lottery-funded charity which provides behavioural interventions to children with autism. So after this introduction, I want to let you know that a recorded version of this webinar will be available for viewing later on the SRF website. And at the end of the presentation, you will all have the opportunity to have your questions answered. We would love to hear from you, so please write your questions in the Q&A or in the chat. And for those of you who are just joining in now, welcome to our talk today by Dr. Andrew Stanfield, entitled Cognition, Behaviour and Clinical Trials in SynGAP 1. It's my pleasure to turn things over now to Dr. Stanfield. Thank you.
Cool. Thank you very much for that introduction, Jess. It seems like such a long time ago I did some of that stuff. It's making me feel old. So I'm going to try and share my screen first of all, and hopefully that will work. You can let me know if it doesn't. Yes, so thank you very much. It's a real pleasure to be invited to speak to you all today. Good morning to those of you who are over in the US, afternoon and evening to those of you who are on my part of the world. As Jess says, my talk's entitled Cognition, Behaviour and Clinical Trials in SynGAP 1. In fact, I mean, what I'm going to do is I'll first of all tell you a little bit about what we do here in Edinburgh, just about the kind of setup and things that we have. I'll then talk a bit about clinical trials in general, you know, including a little bit about trial design and some lessons that we've learned from another condition which I do research in called Fragile X syndrome. And then I will talk about some of the work that we are doing in SynGAP 1 and plan to do. So, first of all, just declarations of interest. For openness sake, I have received grant funding from drug companies in the past for trials in Fragile X syndrome, as well as consultancy fees that come to our research centre from these companies. I should also say for trials in autism as well. So I work at a thing called the Patrick Wild Centre, which is at the University of Edinburgh in Scotland. The Patrick Wild Centre, as this kind of subheading says, is a kind of research centre which is set up to investigate autism and intellectual disability. And we had a particular focus on Fragile X syndrome at the beginning, but we are much bigger than that. And our focus is primarily on monogenetic causes or single gene causes of autism and intellectual disabilities. This picture really is just to demonstrate that families have been key to our centre from the very beginning. The way it all started back in 2008 was a family raising money for a colleague of mine, Professor Peter Kind, who is the other director of the Patrick Wild Centre with me. And that really set off an initiative within the University of Edinburgh it was spotted that this was something that there was potential for, you know, real advances to occur in this field of single gene conditions. You know, genetic science and lab science had moved on, but one of the things that was holding it back really was this silos within universities, research institutions, sort of, and what the idea of the Patrick Wild Centre was to try and bring together people working like myself, who are clinical scientists, with folks working in laboratories like Peter Kind and his group and lots of other people around the university, to really try and advance or to move things forward. The centre itself is named, the picture at the bottom here of the people standing around the plaque is Alfred Wild on the far left, and the centre is named after his brother Patrick, who had autism and who died many, many years ago, but in honour of his parents, who really provided so much care and help for Patrick during his life. At a time, and I know this will resonate even now, when there was very little care or help available for people. You know, there's been a few major landmarks on the way. The opening of the centre occurred in the end of 2010. In 2013, one of the a sister centre, which Professor Peter Kind is a part of, opened in Bangalore in India, and a lot of laboratory science, collaborative laboratory science, is done alongside them. And then in 2015, Dame Stephanie Shirley, who herself had a child who had autism and epilepsy, she donated money to us to open a series of imaging laboratories, microscopic imaging laboratories. And then in 2017, there was a large grant from the Simons Foundation Autism Research Initiative to open a centre called the Simons Initiative for the Developing Brain. And that is something which I think takes up a lot of what we do and encompasses a lot of what we do here in Edinburgh. Apologies to those of you who've heard me talk before, because some of this will sound familiar, but this is SYDBI, which is a collaborative centre within the University of Edinburgh. It's headed up by Peter Kind, who is up on the top left of the screen. And it's primarily built around laboratory science understanding of the biological mechanisms underlying autism. But we have a particular focus on single gene conditions, and almost all of those single gene conditions, in fact, all of them that cause autism also cause intellectual disability. And SynGAP 1 is, Fragile X was probably the first condition that was studied here, but SynGAP 1 is probably the next one along. And there's a lot of people in this picture who've done a lot of work in SynGAP 1 relevant to it. People like Seth Grant and Noboru Komiyama, who were very involved in developing one of the first mouse models of the condition. Peter himself, along with David Wiley, have done a lot of work looking at some of the biochemistry underlying and electrophysiology of models of SynGAP 1. David Fitzpatrick, who was one of the lead investigators of the Deciphering Developmental Disorder study in the UK, where a lot of people who we have met have got their genetic diagnosis from. And then other people here like Stuart Cobb, who's looking at potential genetic rescue for SynGAP 1, Alfredo Gonzalez Solsar, who's doing a lot of EEG in animal models of SynGAP 1. So I suppose what I want to get at, each of these is a PI, a principal investigator, who runs a research group. And not all of them work in SynGAP 1, but a fair number do. And it is, I suppose what I'm trying to get across is that it's a condition of real interest here in Edinburgh, and there's quite a lot of research. I believe Peter is trying to arrange, or you are trying to arrange a date for Peter to come and talk about some of the laboratory science work that his group are doing, which is good because I don't understand all of it, or most of it. What SYDBI aims to do, it's a sister centre to the Patrick Wild Centre. It's about delivering a collaborative centre to look at these biological mechanisms understanding autism. And it's really looking at critical periods when treatments might be most effective, and whether or not, there's a lot of different single gene causes of autism and intellectual disability, but look at the pathways that these genes are involved in, and then to look at, well, does that mean there's convergence of pathways, that there's actually lots of different genes, but they converge on single, a few mechanisms. And that would then mean, if they do, that there might be treatments or interventions that would be, would be useful for more than one cause. It's also about stratifying and kind of just understanding autism spectrum conditions. And then the bottom one, which is the part that I'm primarily involved in, is about thinking about the next steps along moving forward experimental medicine clinical trials for people affected by these conditions, because that's ultimately what all of this work is designed around, and it is hoped to lead to. So, as I said, I'm going to start off by talking a little bit about clinical trials in general. Apologies if this is all just very well known to you. But I think it's useful to go through because it has relevance for some of the things and challenges and things that I'm going to talk about later on in the talk. So I think most people will probably know that there are a number of different development stages for new medicines, which run through from preclinical work in the lab through initial safety work, and then gradually bigger groups looking at safety again, and then moving into, well, whether the drugs actually work. Do they show the same promise in the clinic that they did in the preclinical or the laboratory stages? Now, this is for new medicines. This is for things that are being developed, for example, by the pharmaceutical industry. We're also interested in medicines that are already available, a thing called repurposing of medicines. So where actually the preclinical and phase 0 and phase 1 studies have primarily been done, and then it is a matter of looking at the phase 2 and phase 3 studies. So that skips out a lot of the kind of early development stages if a medicine's already available but maybe being used for something else. But those are, that's really where the two different sources of potential new treatments are going to come from. Again, this is fairly straightforward, just so people are aware. I mean, there are different types of trial design. These are two of the most common, really. Open label studies are studies, they are not randomized, they're not blinded, so everybody who is in the study will get the active medicine, and everybody knows what they are on. So both the person giving the medicine and the people taking the medicine are aware of the medicine that they're on. And they're used very early on in sort of safety studies, or to get some idea about whether or not the medicine might work. If you don't get a signal that it might work in a study where everyone knows that they're taking it, it's very unlikely to work as you move further along. So they're kind of early phase studies, or also sometimes late phase studies. So studies where you're actually looking at after you've done your double-blind trial and people are continuing to take the medicine, looking at longer-term safety or longer-term efficacy or effectiveness. The kind of more common or later phase drug trials tend to all be what we call randomized, double-blind, placebo-controlled trials. So it means in its simplest form, you get two groups and half of them take the placebo medicine, the dummy tablet, half of them take the active medicine. You measure something at the start, and then you measure it again at the end, and you see, well, the people taking the active medicine are doing better than those on the placebo. And the idea is that nobody knows who's on what, so you can't bias it by knowing, oh, well, they're on the active medicine, so they're probably doing better. These are the kind of trials that are required to approve drugs. So, a drug's unlikely to get approved, or very unlikely to get approved on an open label study unless it has truly wondrous results, which has potential for some of the genetic therapies, potentially, I think, in some conditions. Double-blind placebo-controlled trials can either be two-arm studies or they can be crossover studies. So again, people may know this, but in a crossover trial, basically everybody gets the active medicine at some point, but one group get it in the first part of the study, then there's a bit of time while the other group get placebo, then there's a bit of time in the middle called the washout where no one's on any medicines, and then they swap around. So in the second part of the study, the people who got the active medicine in the first bit are now in the placebo, and the people who are on the placebo in the first bit are now getting the active medicine. And there are popular studies for sort of what I would phase 2 trials, because you need fewer people to do them than the other type of design, and they are quite powerful in that everybody acts as their own comparison. So you can compare everybody, you know, who's on the active treatment to what they were like when they were on the placebo. Disadvantage of them is that they take longer to do than other studies, and they are also for medicines that have what we would call a carryover effect, they are not suitable. So if your treatment at time 1 or period 1 is very good and lasts beyond the washout, so if it has ongoing effects, then that interferes with the way that you can analyze the data, because it biases or it affects the findings when you're on the placebo afterwards. So they are useful medicines, but they are useful trial designs, but there are some medicines or approaches that they're not suitable for. This is a much more simple, in some ways, design: active treatment versus placebo. As I said before, measure everyone on the active treatment at the start for whatever you're thinking the drug might help with, and then measure them again at the end. And you hope that your active treatment performs better than those who are on placebo. This is important, and I think a lot of people maybe aren't necessarily aware of this unless you're thinking about and reading about clinical trials. But we think a lot about outcome measures, and outcome measures for clinical trials are a fancy way of saying, what do you think your drug is going to do? So what do you think your medicine is going to help with? So it's usually divided in a primary outcome, which is that is the measure which the whole trial will be judged upon. If you meet your primary outcome and you show benefit to your primary outcome, then you've had a trial that works. Secondary outcomes are kind of exploratory things that you might measure that you think it might help with, but you're not quite so confident, or things you're interested in, and they can help inform future trial designs or other things. But if the trial doesn't, you have to say before you start your study, what's your primary outcome? And then you have to meet that primary outcome to show that your study has worked. So you have to pin your flag to the mast right at the very beginning. And as I'll talk about later on, that's quite complicated for the kind of conditions that we work in. So what do we need to run a successful trial? So then by successful, I don't mean one where we recruit enough people and we get a result of some form. I mean one where hopefully we have a medicine that actually works at the end of it. There may well be other things. These are just things that come to me that are pretty essential. We need to know that the medicine from the laboratory treatment, or the laboratory models, we need to know the medicine is going to be safe, obviously, and we need to know that it is going to work, or at least has it work in the laboratory models. So, and that is quite important, and we will talk about that in a bit. So but it needs, if it doesn't work in your laboratory models, then it's not going to make it out of the lab. So it needs to be something that seems to hit what we want it to hit, and it seems to be safe when people are doing studies in labs. Then we need to know it's safe in people. Those are some of the early phase studies that get done. And then these are some of the big areas that we're interested in, that if it works in the lab, does it then work on people? How likely is it that those treatments that you think work great in your various laboratory models, how likely is it that they're going to work in people as well? Because that's ultimately the key. And then it needs to be the right trial. So you can have the best treatment in the world, but if you don't have enough people in your study, your study will not work. So, all clinical research and all clinical trials live and die by the number of people that they can recruit. It needs to be the right group of people, and we need to measure the right outcome measures. Because if you measure the wrong thing, it may work on it, but if it's not the right primary outcome, then your trial's not going to have been successful. And then I think we need to make sure we actually have the right therapy. It's possible that something might not work by itself, but it might be something that might be helpful if it's combined with either another medicine or potentially a non-medical therapy or non-medication therapy. And the reason I want to talk about this is the sort of cautions from Fragile X syndrome, which I spent quite a while at the beginning of when the Patrick Wild Centre started, involved in these Roche and Novartis studies to look at medicines for this condition, Fragile X syndrome. So if people who know may not know that Fragile X is another single gene condition associated with intellectual disability, commonly associated with autism and ADHD. And it's been around, known about for quite some time, and it has been investigated quite, in some ways, quite thoroughly in the laboratory. And there was a lot of excitement when I started in this post about these clinical trials, you know, that there was a lot of labs around the world had shown that, you know, this particular type of medicines were beneficial for the laboratory models, the mice models primarily. But unfortunately, after several years of doing clinical trials, neither of the drug programs, came to being. And I quite like this back of the envelope calculation by Mark Baer, who works at MIT, and this is taken from the Fraxa website, where he basically calculates that given an 80% chance at each of these steps that you've got it right, what's your overall chance of having a successful clinical trial? If you get the right target and the right medicine to hit that target, you got to get the right dose, give it for the right amount of time to the right people who are at the right age, have the right amount of people, choose your outcome measures correctly, and then it needs to also be acceptable to the people who are taking it. And if you choose 80%, kind of generous 80% at each of those steps, you still come down to about 13% chance of a trial working at the end. So I don't want to be too negative though, but it is one of the things, it always strikes me whenever we see there's a lot of laboratory work that goes on and we see potential therapies that may be helpful, that to bear in mind that there is quite a difference in the steps from things happening in the lab to things happening in the clinic. However, like I said, I don't want to be all negative, because just in November, there was phase 2 Fragile X clinical trial of a medicine that has been through this process, that is, as described in this press release here, unequivocally positive phase 2 results, which didn't require looking at subsets, looking at additional analyses. Their primary outcome measures were met. Really positive news. So although the studies that we were involved in, 10 years ago didn't work, there are other targets, other studies that go on in conditions which then do lead to positive results. So yeah, I was going to talk a bit more about the bottom two of these, about translation and about the right clinical trial. I won't labor the points too long, because I do want to talk about what we're doing in SynGAP. But first of all, translation. So what is translation? I think it's a term most people are probably familiar with. Broadly speaking, we think about it as being bench to bedside, and technically it is bench to bedside and back again. So it is you taking findings from laboratories into clinic, but also taking findings from clinic back to laboratory. And translation is probably the single biggest step where treatment, development of new treatments fails. Some of you may have seen when they were talking about COVID vaccines and the early animal data from COVID vaccines, this phrase, which has been around for a while, I don't know who said it first, but mice lie and monkeys exaggerate. So this is it's a long-standing phrase about how comparable findings in laboratory models, the mice or the monkeys, are to those that come up in humans. And people will often say it's very easy to treat a lot of conditions in mice, much more difficult to treat them in humans. And I think it's particularly difficult, that process of translation, for neuropsychiatric conditions, where there are such dramatic differences between the brain in a human and the brain of a mouse. There's a lot of the basic understanding and how nerve cells work and other cells in the brain work is the same, but the human brain is clearly much more complex than a mouse brain, or even than the brain of other primates. So I think it is particularly difficult for neuropsychiatric conditions. So what are the sorts of things that we are looking at that might help this process? So, a lot of translation depends on looking at really animal behavior or some very sort of basic say nerve structure or something like that. So, it's that we take a look and say, okay, we've given this medicine to these animals. These animals are, you know, they're a model animal of, say, SynGAP1. We give them this medicine and their behavior changes, and we think that is, you know, improved social behavior in the animal, or that is less repetitive behaviors in the animal. You know, but animal behavior is quite complex in a different way from human behavior. It's not clear how much these kind of tests and things actually do translate across into humans. And so what we are interested in, and other groups, is, you know, are there things that get more at brain function that we can compare between species? So we are looking at a few different things. We're looking at cognitive tests, which you can administer in both animals and in humans. We're looking at EEG, so you can look at actual electrical function of the brain in an animal model of a condition, and we can do something similar in humans. And we're looking at MRI, so actually looking at how brain function at a deeper level than EEG is good at looking at the brain surface. fMRI, functional MRI, is better at looking at the kind of deeper parts of the brain as potential ways of translating. The idea being overall that if you can show, first of all, that, you know, say you have a rat, which is what we would call a SynGAP knockout rat. So it's a laboratory model of SynGAP1 related intellectual disability. And you can show that your rat has got cognitive changes, or changes on EEG, or changes on the MRI that are parallel to those seen in humans with these conditions. It gives you a bit more confidence that your rat model is modeling brain function in a kind of more accurate way, or in an accurate way that it is actually a model of the human condition. But then you can look and you can give medicines, say, to the rats, and look for changes in these biomarkers. And if you can then give medicines to humans and see comparable changes in these biomarkers, it gives you more confidence that people's brains are behaving in a similar way, in a comparable way. And that's the idea, that it helps the process of translation. This is just some work done in that. So this is in Cognition, which is probably where it's been most advanced. This was done by Seth Grant's group, along with people who work with me, Andrew McKechnie, and looking at a condition called DLG2, so people with a deletion in this gene called DLG2, and looking at mice with a deletion in this gene called DLG2, and looking at how they perform in this cognitive task. With the mice, actually, they come and they touch this cognitive task, and it's a measure of learning and of what we call reversal learning, so learning different associations, really. And what they showed was that they show comparable difficulties in particular cognitive tasks. So that's the first kind of step here. The next step would be to give medicines to both groups and to see whether or not you show any kind of rescue of these cognitive tasks or improvement in these cognitive tasks in the mice and the humans. And that then would make you more confident that perhaps a longer trial in humans would yield real meaningful clinical effects. Cognition, as I say, that this is the area that has been most advanced. We have these findings. Noboru Komiyama, who I mentioned earlier, along with Seth Grant, has been doing these studies in the SynGAP mouse model, and we are in the process. We are preparing to roll it out into people with SynGAP1. The pandemic has obviously put a bit of a spanner into that work, but also making sure that the tasks who are developed in collaboration from people from another university are working things in people effectively. So we're also looking at EEG and SynGAP1, and I'll talk about that later on. We have done some really nice work in MRI, using MRI in Fragile X, in both rat models of Fragile X and in people. I don't have that data to show you today because it's not fully complete, but it's pretty positive and shows what we think it should show. We've not been doing MRI in people with SynGAP1 because it's probably too difficult to do in everyone, and I think that's probably a next step along for us to look at whether or not it's possible for us to do functional MRI in the group that you guys are most interested in. So the other thing which I said that we need to run a successful trial is having enough people, running the right trial. So having enough people, the right people, measuring the right thing, and thinking about whether or not we are actually testing the right medicine. Do we need more than, do we need to combine therapies? So when I say enough people, we're talking really about participant recruitment to studies. And I mean, I can't emphasize enough, and I say it every time I give a talk, really, you know, without people taking part, studies will always fail. Because if you don't have enough people, you're not going to show a meaningful effect. And I feel this is a very obvious thing to say, so apologies, but recruiting in rare conditions is difficult. You know, and that's why we need things like patient registries and, you know, where people who are able to take part, because these things are not easy to do, and we appreciate that, but where people are able to take part, do register so that they are, the researchers can actually get in touch with them. I think recruitment is particularly hard, again, in the kind of conditions we're talking about, because of the inherent features associated with the conditions: anxiety, difficulties with new experiences, you know, the amount of care and support that people might need to travel places and so forth. So we did a piece of work looking at why people do and do not take part in clinical trials. And this was run by a woman called Sarah Ely, who is our clinical trial coordinator here at the Patrick Wild Centre. This was done in Fragile X, and it was just published this year, but I'm presenting it here because it has, I think it's unlikely to be much different for the SynGAP population. What she was really wanting to do was look at why is it that people don't take part in trials? What are the concerns? What are the barriers to people taking part? And then what are the things that actually might help people take? Why would they take part, and what things might help people take part? And perhaps unsurprisingly, you know, the largest sort of group of reasons why people might not take part is that they're worried about side effects of medicines. They're worried about taking a new medicine or a medicine that's not currently available for the condition they have. So these kind of, you know, safety concerns absolutely are going to be paramount for people. Other things, and what people said when Sarah, so she sent a survey to about 200 people, and then she did focus groups with a number of folk. And what people said was that, you know, there's acceptance that any medicine carries risk, but it's about making sure that they are given enough information in a way that people understand, with as much safety data as possible. So an openness, and that does happen, but I don't know if anyone's ever taken part in a clinical trial, but you often get information sheets with about 20 pages in them. So I think we need to be clear, you know, and that's too much for anyone to read, really. So we need to be clear, very clear in the information we give, and upfront with people about what the potential safety issues, side effects or other safety issues are, in so much as we know. Other reasons why people might not wish to take part could be divided into sort of trial procedures. So blood tests, swallowing tablets were two big things. You know, and we talked a lot with people about thinking about desensitization, minimizing the amount of blood testing, thinking about, is there ways that we could get people ready or help them to be able to have blood tests more easily. And then looking at different ways that we can provide medicines, you know, accepting that tablets are hard for people. So looking at liquids or powders that can be scattered over food. And then the other big reason, and this is one of the other main reasons people didn't, you know, were worried about taking part, was about the effects on their wider life. So having the, you know, financial and time effects, having to travel was a big one, and that was both a combination of the time that it took, but also the difficulties. I probably do not have to tell anyone who is listening to this about some of the difficulties that people have in traveling to, even locally sometimes, but particularly if you're traveling far away to a new place where you've not met the people before and where your child has not met them before, that can be pretty anxiety provoking. So, you know, one of the things that we are considering and are likely to do, and I think the pandemic actually makes this sort of thing more easy to do is to look at, well, actually, can we really reduce the number of times people would need to come and see us? You know, try and get other sites that might be local to people, but also say, well, do you really need to come all this time, or can we do some of these by video? You know, and look at doing remote visits. Which I think, as I say, all of the move to using Zoom and Teams and so forth in the pandemic is likely to make that easier and more acceptable to people. The next thing, and I'm not going to talk a lot about this, but it's about having the right group of people. I don't think we know in SynGAP 1 what the right subgroups are. I think there's of people that should be coming into clinical trials. There's an instinctive feeling I think we all have that medicines are likely to be more effective if they're started in younger people. You know, but there is some laboratory evidence from Gavin Rumbaugh's group in recent times, you know, showing that there are still improvements that can potentially be made in young adulthood. These were done in mice, not in people, I should say. But you know, that it's not necessarily that if people haven't been treated when they're very young, that they may not be able to still show improvement. And I think, to be honest, what age needs treatment should be started is going to be different between different genetic conditions, between different mutations, potentially even within the gene. And certainly depending on what the actual treatment. Some treatments might work in older people, but they don't work in younger individuals because they target different circuits that are you know, maybe more amenable to change in older people. And it's probably going to be symptom specific as well. It's quite possible that improvement can be made, for example, in seizures or in some other symptoms in adulthood, whereas, you know, it may be harder to change other symptoms. But we really don't know at the moment. I suppose I would say it's important not to generalize them. That's probably my main message. I think we need more work in older animal models, because a lot of work is often done in very young animal models. So I think we need work in older animal models to look at you know, whether or not treatments are effective in those. And then I think we need to do trials across the age range in people. And I think this comes from my experience and lots of our experience in Fragile X, that, you know, if a medicine doesn't work in an adult or an adolescent, it's not a reason to stop studying it. It's not a reason not to look down to younger individuals, because it might be that it might be effective for them. So it's important not to generalize too much when thinking about age and so forth in clinical trials. And we need to measure the right outcome. And I've put this, including biomarkers, with a question mark here as well. I just want to talk a little bit about outcome measures generally. So, I mean, this is something all of you will have to have done, getting your blood pressure measured. I know it's not as simple as this. I have cardiologist friends who tell me that this is not 100% correct to say, but sometimes I wish I was doing a trial of a medicine to lower blood pressure, because you can get an objective measure at the very beginning that is, you know, a number, and you can do that in all of your people, and then you can measure it again at the end of the study, and you can see if those on the active treatment, if that number is lower in those in the active treatment than in those of the who took the placebo. And the nice thing about that is that we also know, again, it's not quite as simple as this, but we do largely know that if you lower blood pressure, you lower the risk of future cardiac events. So, in other words, lowering blood pressure in itself, I mean, if we could get a drug and we could show it lowers blood pressure, the only reason that's worthwhile is because it has an actual real clinical meaning for people. You know, so it means that later on they're less likely to have heart attacks or strokes or whatever. So it is a biomarker, you could say. It's a you know, quantitative measure of a biological function that is in itself not that meaningful, but is a meaningful predictor of future health. So, you know, there's something that's really appealing and really attractive about something as nice and simple as blood pressure as an outcome measure. Obviously, we don't have that in neurodevelopmental conditions. So, again, this is coming from Fragile X studies, that a lot of outcome measures that people have used and continue to use are based on observer-reported behavior. And that's, I mean, that's understandable, because ultimately, at the end of the day, for a treatment to be meaningful, it has to make an observable difference to either the person themselves or the people round about them. But the problem with these measures is that they're very subjective, and there's a large kind of placebo effect associated with them. So I just put this graph in just to, you don't really need to worry about what each of the lines is, but this was in some of the Fragile X trials. And the key thing here really is that for the first 4 weeks, the bit at the very left-hand side of that graph, everybody had placebo. And in that first 4 weeks, on average, pretty much, well, not on average, overall, pretty much everyone improved. And some people improved quite dramatically. You know, the scale on the left-hand side, -20, is a really significant improvement. You know, that's a successful treatment, you know, in terms of that scale. So, you know, and that is when people were on placebo. And I should be clear as well, I mean, I've not got time really to go into the placebo response. It's not like it's made up. It's not that somebody is like, oh, I think they're better, so they kind of are. I mean, people do show improvements that occur just by taking part in a clinical trial, by coming along, by seeing people, for some families, some of the trials that we've run, you know, they're having to do new things. So they come out of the, you know, they get a flight to come up and see us in Edinburgh. They get the train up in Edinburgh because they want to come to this trial. And that then starts to open up the world a bit more, and the people feel more confident. They're, you know, the patient or the affected person himself feels a bit better about doing some new things. So there are real genuine benefits. It's not just a kind of made up effect. But it is an effect that's non-specific, and it's not due to the drug. So this is one of the biggest problems with outcome measures in these kind of trials. And so we need better ones, or additional ones, really. So, I mean, this is just a list of things that are good outcome measures should have. They need to be something that's acceptable, that we can measure and people will like. It needs to be something that captures impairment, that is meaningful to people, or it marks something meaningful. So like the blood pressure measure that I talked about. It needs to be something reliable so you can repeat it and get a similar kind of finding. It needs ideally objective. So something that's not going to be as vulnerable to placebo effects. And then it needs to capture changes over time. So again, the sort of things that we're looking at are the sort of things that I talked a bit about earlier on as potential translational measures. So measures of cognition, measures of brain function. Both, you know, the top is doing those touchscreen tests. The little picture below is a child doing some eye tracking tests of cognition, which are really nice because they don't have to involve the baby, in this case, actively pressing buttons or doing anything. They're passive tests, but they can measure cognitive function. And then things like an EEG or functional imaging are also potential outcome measures. May also be potential biomarkers that might, you know, predict change. I'm not sure that we'll ever have clinical trials where we give someone a medicine and then we take their EEG and we say, oh yeah, cool, it causes a change in the EEG signal, so there we go, this medicine works, off we go. What we might have is trials where we might see changes in an EEG signal early on in treatment, and that would then motivate us to continue on with treatment with the medicine. So it might be a way of helping to pick out those, as I say, predictor of future response and of clinically meaningful benefit. The other thing, I'm not going to labor this, is, you know, one therapy sufficient. You know, one of our families who's involved with us just talked about clinical trials once as being like, it's a bit like teaching someone to read in the dark. So you need to turn the light on, but then you also need to teach them how to read. So a medicine might turn a light on, but you may also need another intervention, a learning intervention, a language intervention, a behavior intervention that will then interact with that medicine to make the biggest kind of differences, you know, particularly over short periods of time. So there's a few good examples. This is increasingly common, and there's a few good examples in Fragile X, for example. Fragile X syndrome, but there is also in other conditions as well. And I think if we want to try and maximize benefits, then this is a good way to go. So this is probably what you're all thinking. I'm aware it is now 5:15 here, so I'm running later than I had intended to. And you know, I've yet really to talk about what we're doing in SynGAP 1. So that's what this part of the talk is going to be about. I'm hoping that you have stayed on for it. I can't see how many people are still listening at the moment, but hopefully you're still there. I presented.
We have about 27 people still on.
Oh, excellent.
There.
Oh, good. It's always reassuring. It feels slightly strange doing a presentation this because you feel like you're just talking to yourself. So yes, what are we doing in SynGAP 1? And some people here, indeed some people on the call will have participated in some of this research. This is one of the big studies that we're running at the moment, which we call the Neuro-GD study, Neurocognitive Phenotypes in Monogenic Developmental Disorders. Basically, it's a study where what we're doing is looking a bit like I talked about some of the aims for SIDB earlier on about convergence and divergence, looking at, well, what conditions have overlaps between them and what ones have differences. And it comes from this paper that David Wiley and Peter Kine's group did about 5 years ago now, looking at SynGAP, their SynGAP mouse model and the Fragile X mouse model. And what they found in that paper was that there are overlaps between these groups in terms of how the biochemical pathways in the hippocampus, so the electrophysiological path in the hippocampus, there was a high degree of convergence between what was working differently in your SynGAP mice and your Fragile X mice compared to typically developing mice. And then they also showed in this study as well that the medicine lovastatin reversed these changes in the hippocampus in both of these mice. And the hippocampus, as people will probably know, is an area of the brain particularly important for things like memory and learning. And so it was, you know, quite exciting because A, it showed that there was similarities and convergence between these conditions, and B, it was also, I think, the first study to look at lovastatin or any intervention in a mouse model. I may be wrong with that, but certainly lovastatin in a mouse model of SynGAP. And that's what kind of motivated this study. So we are really, talks about monogenic developmental disorders, and it's set up to look at as many as we wish to, but at the moment we're really only looking at Fragile X and SynGAP 1. And it is, like I say, A, to look at convergence between the conditions. Do we see that overlap happening that we see in the mouse model? But B, also to fulfill that other thing I mentioned earlier on for the Simons Initiative, which is understanding these conditions, deep phenotyping, as we call it, understanding these conditions at a level which will be helpful for running clinical trials. Now we know a lot of this work has been done for Fragile X before, so the most novel part of it is looking at characterizing people with SynGAP 1. So this first group of questionnaires and interviews are all things that are sort of clinical outcome measures to look at some of the kind of behavioral traits that we, people with SynGAP 1 might display. There's a measure of nonverbal intelligence as well. And then there is these measures which are more of those kind of cognitive or brain function measures. So looking at cognitive touch, those touchscreen measures, looking at eye tracking, as I said earlier on, a test of how the brain functions, and then looking at various different EEG kind of signatures of cognition and connectivity. And then particularly this group are also translational measures, that we can do both in the humans and we can do them in the laboratory model. So it's hitting a few different important aspects which are going to hopefully help us move more towards clinical trials. So I'll talk, I presented some of these data fairly recently, so some of you may have seen them at another conference. We actually have now 28 people with SynGAP 1 in this study, but the data that we have got, complete data and all organized and sorted out, is in about 18. almost everyone's under the age of 14 who took part in the study. We have one adult who is 34, but they most people are in the younger kind of age groups. And really, what we're aiming to do here is look at whether or not these measures might capture the behaviors that people present with, so that maybe we could use them as sort of subjective measures that we can use in clinical trials, but also just to try to describe in detail the sort of behaviors that people present with. You guys all know, and families talk about you know, how their children present and things, but actually putting it down in the scientific literature, so it's out there that it can be given to clinicians and so forth, using these kind of standardized measures is also, I think, one of the other potential benefits of this study. So I'll talk about some of these here. One thing I'm not going to talk about, but which I think is probably really interesting, and we are starting to have a look or analysis of it, is what I call a qualitative description of the general behavior. So that's just an interview with families to say, what are the behaviors? What is your, you know, describe your child. What are they like? What are they good at? What do they struggle with? What are the you know, what are the behaviors that cause you the most difficulty? That kind of thing. Because there's something beyond what these kind of standardized measures can capture, which we hope to try and get at with some of those kind of qualitative or personal descriptions from families. So this is measures of autistic traits. The dotted line, which runs up from 65 upwards, is what we would call moderate impairment. This is a screening test called the Social Responsiveness Scale. And if you score above 65, you're felt to be highly likely to, or no, you should move forward for further diagnostic screening for autism. Now, I qualify this by saying that when we speak to families, that certainly folks do describe autistic traits, that people will often say that, you know, it's maybe different from other kids who don't have SynGAP, and that's very similar to what folks see in Fragile X as well, actually, that, you know, there are differences in how these sort of difficulties present, but because our descriptions aren't so good, they kind of often get rolled under this sort of rubric of autism. Our kind of finding papers to date have suggested, you know, 50%, maybe a bit more of people presenting with diagnoses of autism. Certainly our findings would say that there are much greater numbers than that who are presenting with very high levels of autistic traits. Now, whether everyone goes on to get a diagnosis of autism, they unlikely to, but it, you know, when looked at in more detail, it does seem like there is, you know, higher rates of autism than are perhaps described in some of the existing literature, and then perhaps are diagnosed, that people maybe have, you know, quite high levels of autistic traits, but they never actually get a formal diagnosis of autism. Hyperactivity, you know, can come under ADHD, but again, very high levels of hyperactivity, difficulties with attention, and you know, impulsive kind of behaviors that we see. That, you know, and it was one of the, we had a day where we had maybe 13 or 14 families who came along, and it was very clear that a lot of children were very active. Yeah, it's one of those things that I think is quite common. Again, whether it is ADHD or whether it is, you know, different from that, we still don't really know. And I know there's been some, I saw Jimmy Holder present some nice work just looking at which medicines had helped people, and you know, from what they'd seemed to find, stimulant medicines maybe weren't so good, but medicines like clonidine or guanfacine had been quite helpful for some of the difficulties people had. So yeah, you know, that's part of one of the things that we hope to get at a bit more with some of the kind of qualitative interviews with people. But again, I think just very, you know, high levels of difficulty. People score highly on these scales. This is the CBCL, or Childhood Behavior Checklist, which covers a whole load of different kind of domains of behavior. You know, people would sometimes call it difficult behaviors or behaviors that cause people problems. And again, that dotted line is clinically significant. And you know, a lot of our group of people score above this dotted line. And yeah, the people have asked, and I think an area of real interest for us is what happens over time. So, you know, this is a snapshot taken of a group of people between the ages of about 3 and 12 or 14. You know, but how does this develop and change over time? You know, we expect probably autistic traits not to change. We'd expect ADHD traits maybe to get better, but behavior, you know, moving through adolescence is a difficult time for any children and parent. But you know, how does people's behavior change over that time, and how are people as adults? And I think these are areas that we need to know an awful lot more about. We don't have the data at the moment. You know, we don't really have groups of people in those older age groups to look at, but it's something that would be really interesting to take forward. Sleep is something that families talk to us about, and you know, these are data on just an overall sleep questionnaire, and lots of difficulties in sleep, you know, particularly with behavior overnight, and sometimes with not going to bed, not going to sleep at night time. Jimmy Holder and Connie Smith-Hicks also have data with the same kind of questionnaire, so we're going to combine it all together to look at it in greater numbers. You know, but it's definitely something that is an issue and needs investigated more, and I will talk very briefly about that piece of research that we're doing or starting soon, sometime towards the end. And sensory issues, again, very common, particularly in the touch domain. You know, that people have both sensory seeking and sensory avoiding behavior, and particularly have the hypersensitive to touch. A lot of people talk to us about enjoying children who enjoy the feeling of water, you know, but also people who dislike certain feelings. Loud noises is a common one that people dislike. But yeah, sort of very commonly people are presenting with sensory difficulties, which are again part of autistic traits, but may well be you know, have an underlying difference from people with autism with other causes. This is the other thing that one of the other things that we looked at, which is about adaptive behavior. What can people do? Where are their skills? How well do they manage? Adaptive behavior is basically the skills that you acquire to move through day-to-day life. And this test of Vineland looks at social skills, it looks at daily living skills, things like washing, dressing, you know, eating and that, and then it looks at communication skills. And scores of under 70 are said to be sort of significant impairment. And a lot of people are presenting with significant impairment for their age range. Although interestingly, some not. Some people are performing, you know, quite well in various parts of this scale. But I don't think I'm saying anything that people won't know, that, you know, folks' ability and adaptive behavior is impaired. And when we look across age, at first glance, what it looks like is that as people get older, that their abilities get less, but that's not true. That's because of the way the scale is done. So it's not that people are losing skills. It's not that people are regressing and becoming worse at things, but they are struggling to keep up with the skills of the general population. So people still are acquiring new skills, you know, but not at the same rate as those people in the general population. Now, we need to look at this longitudinally. We need to look at this, and as I said earlier on, we have very little data in anyone above the age of 14. So we need to find out more about the adult phenotype, but the way people present as adults. But I suppose my message is that it's not that people don't learn skills, but it does take them longer, and they don't acquire them to the same way that some other individuals who aren't affected do. I'll briefly touch on some of the biomarker stuff. This has all been kiboshed, for want of a better term, by the pandemic, because these require face-to-face assessments. They're not things that we can particularly do over video at the moment, but they are things that we had just started to see people again in November time, and I don't know what the rules are like where everyone else is, but here in the UK, we are back on a total lockdown, so we're not really able to see anybody for research at the moment. Hopefully, as the vaccine gets rolled out, the situation will change. But we are looking at this reversal learning test that, like I say, we have done in another genetic condition before. Our colleagues of mine have done in another genetic condition before. And that it is something that we have got translational measures for in the mice models of SynGAP1. So this is something that, as I say, that's quite well worked up in the mice models, and is going to be of real interest to see, is it a potential translational marker in that works in both, that we see difficulties in both the mice and in humans. We're doing eye tracking, which I mentioned earlier on, as a passive test of cognition, both to look at people's, one of these tests is looking at, focus on social stimuli, as opposed to, we are supposed to look more at faces than we are at other parts of the body or non-human objects. So these heat maps can tell you how long people look at things. I do worry about having a mobile phone, though. People automatically look towards that. But it's to get a handle on social processing, really. And then one of the other tests that we're doing is, you have this group of stimuli, and naturally people's eyes are drawn towards the odd one out. And how quickly you're drawn towards the odd one out is a kind of measure of inspection time, which in itself is a measure, is a cognitive function. And not just how quickly, but also what sort of patterns of eye tracking that you use. So the top one is a much more sort of organized, quickly drawn over to the O, whereas the bottom, the person is looking much more, but probably slightly more at the O than most of the other stimuli, but it's not nearly so organized. So, and these are data that are taken from children with SynGAP1. We just don't have enough to make any clear statements about it. We're also doing EEGs while people are, to look at visual processing, to look at auditory processing, and also while people are at rest. So we're not looking here at seizures. What we're looking at is, how very primary kind of auditory and primary sensory processing and brain connectivity. So it's not so much looking for biomarkers in terms of seizures or spiking, but in terms of differences in sensory processing. These are some of our kind of good quality traces, for want of a better term. As I've said in other talks before, a lot of our traces look like this because children with SynGAP1 find having an EEG cap put on and sitting and watching boring screens flash or listening to tones not very exciting. So there's a lot of movement, and we've recently kind of updated our equipment and our protocols to try and improve this as we do move forward and come back out of the pandemic. And it will be possible. It is just something that not for everybody, but it is going to be possible for some people. But I think it is something that we need to keep constantly refining how we actually acquire the data. I said I would mention this briefly. This is separate from the Neuro-GD study, but this is something that we have just got funded, or a colleague of mine, Dr. Lindsay Mizen, has just got funded. And what she's going to do is to look at detailed characterizing sleep difficulties in SynGAP1 using sort of formal sleep studies. So ideally, assuming that we can do this because of the pandemic, and assuming people are willing to let us, what Lindsay and her team are going to do is hopefully to visit people at home and to monitor over a couple of nights sleep using a device which monitors things like heart rates, respiratory rate, but monitors also a few electrodes for EEG signals and things, to really try and get a detailed handle on what's going on in sleep. In part because there are some sleep difficulties that there are existing treatments for. So it's possible, can we actually diagnose sort of known sleep pathology, for want of a better term? But also this is coordinated with some of that we're also doing, again, I say we, it's a broad we. Colleagues within the Simon's Initiative for Developing Brain are also doing sleep studies in the rat model of SynGAP1. So again, thinking about these translational markers, they're doing various things to look at sleep staging and EEG, heart rate, etc. And thinking about these translational markers that we might be able to use to develop better treatments moving forwards, or better translation of treatments moving forwards. And looking forwards, you know, I've talked before about, the whole idea of setting up what we want to do is to look at clinical trials. You know, and there is some animal data that come from colleagues of ours to, let's say, lovastatin may be effective. More recently, and this is published as a preprint at the moment, Peter Kine's group have shown that the medicine incl, which is an anti-epileptic drug, was particularly effective in the rat models for seizures. And whether, you know, there's lots of people out there who will have had ethosuximide and which may or may not have been beneficial, but whether it's worth us pursuing that in different dosing or possibly in combination with other medicines is something that we're considering at the moment. I'm also interested in what people think. You know, these lovastatins targeted at underlying biochemistry, ethosuximide is targeted at seizures. There's obviously, and I've not talked about this, but there's obviously interest in things like genetic therapies, which are targeted at, you know, real, you know, the underlying sort of core features of the conditions. But there are also medicines out there that are used quite widely in children and in adults for things like ADHD and anxiety, and you know, which are prescribed and given to children, but which with you know, some of your children may well be taking them, but we don't have good evidence for what the best type of medicine is that we should be giving to people. So could we look at doing trials of medicines that are for symptoms, not so much targeted at underlying core features, but actually saying, you know, what's the optimal overall treatment strategy if you're looking at medicines that you should choose to consider someone with SynGAP1 who presents in this way or with this difficulty. I think there is also definitely, and this is not something we are actively pursuing at the moment, but I do think that, as I say, to maximize the chance of success, we want to be combining our drug treatments with non-medication based interventions. And I think, if I'm honest, in the first instance, and this is something that we are talking a lot about here, is looking to do a feasibility study using one of the medicines from the above, or if there is other potential candidates, you know, to say, actually, can we do clinical trials in SynGAP1 for us in the UK, but also looking worldwide, you know, to see about overcoming these barriers. How do we do them? What are the logistics? You know, are people able to come? Can we do the things? Can we do a lot of it remotely for medicines that are known to be pretty safe to take? So, you know, actually looking forward and saying, you know, the first step is to do a feasibility study of our best candidate, open label trial, and to see, does it, you know, does it help? But also, as I say, is it possible? Because I think if we you know, we need to start with small studies that show that we can then move on to do bigger studies. So I think that is it. This is just the various funders, people we want to thank. That's our the Bridge the Gap SynGAP day, family day that we had in Edinburgh in 2019. 2020's was cancelled, but we have a date for 2021, which was attended by at least some of the people on this call. This is the most up-to-date photo of my specific research group. This is our Christmas card this year, which we could only do socially distanced by Zoom. Lindsay Mizen's up in the top left of that. She's doing the sleep study. I'm the terrible photo on the top right. Ashling and Damien, who are doing a lot of the Neuro-GD study, and who some of you will have either met or have met by Zoom over the last while. Andrew McKechnie, who's done some of that work that I showed with the translational cognition, and who's been involved in a lot of the clinical trials. And then Sarah Ely at the bottom, who is our clinical trial coordinator here in the center. These are the people who've done most of the work that I've presented and will be taking things forward. So thank you. I am happy to hang around. I'm aware I've gone over my time, but I'm happy to hang around and take questions if people have them. I'll just try and stop sharing my
we do
screen.
have a couple of questions in the Q&A. They're coming in. I just wanted to say first, thank you very much, Dr. Stanfield, and to your whole team for the work that you do. And you clearly summarize the challenges that we have in developing clinical trials, particularly in approximating the efficacy between animal models and the humans, and the challenges in finding biomarkers. You also spoke about the barriers to recruitment and also the challenges that the families face in, being able to participate. So we appreciate that. So I will start with the first question, which was, what is generally the minimum number of participants required for a trial? So
it depends. It depends what type of study you're doing, what type of trial you are doing. For a definitive study, for the kind of study that, say, the Fragile X trials, for example, that we were looking at, that we were involved in, that we're looking to see about potentially getting a medicine license. Those were studies which had, 150 plus people in them, which is big for our fields, but small compared to cardiology studies and those sorts of things. It depends how effective your treatment is likely to be as well. If you've got a really effective treatment, then you don't need that many people to show that it works. I would say if we were doing, and we are talking about this at the moment, for a feasibility study, we'd start off with maybe 10 or 15 people in an open label study to show that, this might work, get some idea, signal of potential benefit. I think then if you're moving into a kind of crossover study, if you can get 30 or 40 people together, and these are, back of a envelope calculations, that would, have a reasonable chance of success. But you do need to get into the hundreds to really, I think, prove effectiveness in a way that, say, drug regulators are going to be happy with. The one big caveat to that is, if you're doing some of these genetic trials or genetic rescue studies that are done, where they've got very massive effects in conditions, some neurological conditions in very young children, you don't need those sort of numbers. So, it depends on how effective you think it is, and also what you think it's going to do, what you're actually testing for people. I see Mike has his hand up.
Jess, can I throw a couple questions in?
Sure.
Sorry. Two biggies, Dr. Stanfield, and of course, first, thank you. That was comprehensive and very helpful. Fragile X, I think, is an interesting one that I've come to appreciate. A lot of our patients get, when the younger kids present, the clinicians are more familiar with Fragile X because it's been around longer, better understood. And some clinicians, including my own, test for Fragile X. And then they get a negative, and my clinician at least was like, okay, it's not genetic, which was obviously a silly thing to say. And that's why they're no longer my neurologist. But I think it's helpful for the community to understand, like, the Fragile X, and I'm just going to say this and invite you to correct it. Fragile X is a much bigger population, much better understood. And very often our kids, that's the first suspicion, but then good neuros generally keep going with the testing. So phenotypically, that's interesting, right? We have a lot of overlap with Fragile X. Fragile X has been around much longer, has spent much more money. Question one, assuming everything I just said was directionally correct. Question one, how can we benefit from all the money that Mike and the team at the Fragile X Foundation have spent on that work? And then getting inside the body, why is it that our kids look like Fragile X? Is it that all kids with NDDs kind of look the same in the early years? Or is there something there in your overlapping expertise between those two syndromes that what is the relationship between SynGAP and Fragile X? Because it keeps coming up. So that's my first question, and then I have another one.
Okay. So the how to benefit from the work that has been done. I mean, I think some of what I talked about in terms of trial design, I think the Fragile X field is probably fairly high up in terms of the way it's thinking about running trials in children. Combining them. The Forward study, which is being run by Liz Berry-Kravis at Rush, is looking very in young children with a good length of time of treatment, and along with a kind of another intervention as well. So even the sort of the failures in Fragile X, I think, have led to us having a better idea about how to do trials. In terms of, they're actually understanding they affect overlapping kind of pathways basically in the synapse. Which if you look at these diagrams of the gene that affects Fragile X, FMR1 and SynGAP, the proteins at the synapse in the brain, they all, I wouldn't go so far as to say they form part of the same pathway, but these pathways overlap and they cross over and they converge, which is what motivated Peter's group to go and look for that convergence in animal models. And it's actually a big part of what motivated looking for convergence across other models as well. So, I think that there's the work that's been done in Fragile X points somewhat to potential things that might be helpful in SynGAP. That was why people tried lovastatin. It had been shown in Fragile X, and then that was one of the reasons to look at it in SynGAP in that Barnes paper that was done here five years ago or so. So, I think I think there's some learning that can be done. I don't want to overstate the convergence though. I think part of the reason clinicians always look for Fragile X is because it is one of the more common genetic findings. It's been known about for a long time. I say people are familiar with that. And there are definitely features which overlap. But as you say, good clinicians, everyone should be checked for Fragile X. SynGAP may not be that much less common. It's just much less commonly identified because the tests required are more complex, more expensive, and not as widely available. That's changing. I hear certainly our clinical genetics department that we're thanks in part to studies like the DDD study, where and actually exome sequencing, which does identify a lot of cases of SynGAP, is becoming a second line but available test within the UK Health Service. So it is not. It will take a while, I think, often for people to get it, unfortunately, by the time people go through, as everyone knows, delays, but it is changing. And I think we will see a lot more. I mean, we've already seen more people diagnosed with SynGAP and people diagnosed through more clinics, and awareness should grow. I would say as well that one of the big frustrations in the Fragile X community is that doctors still don't seem to know a lot about it. Most of them go, oh yeah, I remember that at medical school, it causes learning difficulties, doesn't it? And that's, and I think that's, unfortunately, anything that is of even the one in a few thousand rarity, that is often a problem that people are going to meet, unfortunately.
Yeah, and
I wanted to follow up with that as well, because obviously we suffer even more from the underdiagnosis. So I was going to ask you to speak about sort of the barriers involved in diagnosing adults, because as you mentioned during the talk, you have very little data on the adult population, and as a community we know very few diagnosed adults as well. So I appreciate your thoughts on how we as a parent-led advocacy organization can help try and reach some of those people and get them a diagnosis.
Yeah, so this is something I'm really interested in. A lot of my clinical work is actually with adults with intellectual disability. And we work in a specialists center where there's been a long history of interest in genetic conditions, and about 40% of people within our clinical services have had have had some kind of genetic test at some point of their life. So that may mean that they've had a look down a microscope at some chromosomes when they were 4, or something like that. Very, very few are investigated as adults using exome sequencing and looking for these kind of genes, and that's not because it's not available, it's just not taken up by clinicians. And I think there is a hearts and minds operation to be won with clinicians, and I think we are beginning to get there a bit, to say it is useful to test in adulthood. I think there is a and I think this is the wrong view, but I think there is a view from some people saying, well, you know, they're adults now. What does it, it's not like there's a treatment, so what does it really matter? And I think organizations like yours, like it's the explosion of the internet, of, you know, parental support organizations and advocacy groups and things, are changing that conversation to say, you know, actually, even just to have a reason, and then a group that you can connect with and you can hear from other families, peer support, that in itself is a good enough reason. And I do think that is changed, but I do think it is a kind of clinician hearts and minds sort of battle to be won. A study I've not mentioned that we are slogging our way through the permissions required to set it up. It shouldn't be this complicated, but it is. But is to actually start doing exome sequencing in adults with intellectual disability, moderate to severe ID within our population, to demonstrate to people what both that it is, you know, a feasibility study that people want it, families want it, you know, and then to start to look not at how many folk there are with different conditions, but to try and start to see, well, what are the adults? What do people look like as adults? You know, because I think we don't, most of these, like SynGAP and a lot of the new single gene conditions where you've got diagnosed on sequencing, we just don't know. It's all done in children. So I sort of hope, my hope is we're doing a feasibility study in the first instance, looking at maybe 100 people, but the idea is that we will expand that to get the sorts of numbers where we will be picking up undiagnosed cases. We'll pick up plenty of undiagnosed cases of genetic disorders with it, and to start to answer these questions about adult phenotypes. And I think that will help to win this hearts and minds operation from my side. I think there is a parental and, you know, that kind of advocacy work, which is, you know, important, I think, to do with clinicians. How you reach them, adults are often not, they're not in a school service, they're not in education. It's harder to find, you know, but carers organizations and things like that more generally are often places where family members, you know, are kind of are. So, you know, for undiagnosed individuals or other, you know, broader learning disability charities. But yeah, it's, we've been talking about this for 10 years. I mean, but I first had a conversation about trying to increase the diagnosis in adults. It was 11 years ago at a conference locally, and I don't think there's been much difference since then. I think the arrival of exome sequencing will change that conversation now, because that wasn't the case back then.
Jess, can I please say a couple things?
Yeah, sure, go ahead.
I'm actually going to share my screen if you don't mind. I just want to share a couple things with Dr. Stanfield and the audience. So you guys seeing in Google right now?
Yep.
Yeah. So if you go to syngap.fund/incidents, that's my short link. I like links to this article in which I tackled the issue of SynGAP prevalence, Dr. Stanfield, and I walked through in here all the studies, the little cohort studies that have gave us that 1 to 0.5% of ID. And then I talked about Dr. Lal's group's work on his predicted incidence and how it talks about SynGAP. But basically, this is interesting because it suggests that we're radically underdiagnosed in all geographies, number one. And number two, there was a cohort study in Denmark that I'm sure you're familiar with where they looked at 200 and they found one SynGAPian. So that's just interesting. The other thing I want to shout out is in the States, and we're going to invite in April, we have this digital natural history study SRF is funding with Citizen. And this is my personal log-on, right? So what Citizen is we don't, nobody gets on a plane. We just collect patients' existing medical records. And then you and any other researcher would get an anonymized, normalized version of this data. So you wouldn't get the PDFs because those are identifiable, but you would get the full clinical history of every patient who's in the study. It's freely available. The IRB is written very flexibly. And just as an example, I, as a parent, if you called me up and said, let's do an interview, hey, has Tony done a Vineland? I can search, I must be spelling that wrong because it worked 5 minutes ago. I can search for Vineland or whatever, or I can say, yeah, I know he's done a Vineland. He did it at Palo Alto Medical, and I can search for medicines or what have you. But I have Tony's I have every page of Tony's medical record here, including, by the way, MRIs and imaging. And we have about 100 US patients in here, and we're going to invite Brits soon. I'm so bummed out that the Vineland's not working because I promised that it did before. That's how you spell it, isn't it?
Yep. Yeah, it is.
I'll come back. But this exists and it's available to you and any researcher for free. And we have 100 patients. So while you're under COVID lockdown, it might be something you and your colleagues would be interested in exploring. And the last thing, this is not public yet, but I can't help myself. We have found that a 65-year-old patient in the US, and we're so excited about her. We made a brief movie about her life that we'll be releasing for Rare Disease Day on February 26th. So a little bit more collateral in your arsenal around getting people to think about adults, because her life story is quite fascinating. And we haven't announced this yet, but I just want to tease it because you're exactly right. I think part of our job as SRF is to, and we'll do a UK screening with SRF UK too, is to let people know these patients are out there, and just because we don't diagnose them as kids, they're still sitting somewhere, right? And we've got to figure out what happens to them. And I think there are both therapeutic, both drug and services implications once you understand the monogenic cause of disease. And so it's hard to think about my kid growing up and being an adult with this disease, but it's even harder to think about adults with this disease that haven't even been diagnosed. Mm-hmm. So I'm really heartened to hear that you're focusing on that. Okay. Yeah.
It's one of the things. So it is. I mean, I'm aware of the citizen stuff, and it is definitely something that is on our radar. It's one of the things that we would hope to look at as well with what we're doing with the adults is to do what, like, linking data to existing health records as well. Which there are certain. The way the UK had, there are certain health records that are available nationally that we can actually link and look at hospital admissions and things like that. So, because I just think it's something, as you say, we know nothing about it. I think there's adult patients out there who aren't diagnosed, and then whose families also aren't able to get kind of support or things as well, which is, it's a big issue.
All right, I'm just going to pick up another hopefully simple couple of questions before we wrap up. We had a question regarding the sleep study and whether or not your team collaborates with Dr. Alex Baez from Hospital Sant Joan de Déu. I hope I'm pronouncing that correctly, in Barcelona.
Yeah, so, and I see the one below about interchange of information would be reasonable. So yeah. I don't know Dr. Baez. I know him academically. I am aware of him, but I don't know exactly what they're doing. It's helpful to hear that they're also doing sleep studies, because we will get in touch. And I had a video conference with Jimmy Holder and Connie Smith-Hicks to talk about the sleep work that they've been doing as well. We are dead keen on sharing information. We're very open with sharing what we're doing and sharing our data, because we will all get further forward if we work together. So that's helpful. I will send Dr. Baez an email by way of introduction.
Great.
Excellent.
I'll take another question from Katrine Deckers, who's the chairperson of SRF in the UK. And she's asking if there's any positives as a result of the COVID-19 vaccine development that will help inform drug development that we might benefit from.
Oh, I don't know. The COVID vaccine development was so quick because everybody, A, the tools were all there, but B, the regulators went through it immediately and quickly. That's why it was so sped up in that sense. People were writing trial protocols in a few days, and then the drug regulators were approving them in a day or two. Which that six months, a year is a normal process, and I don't think that we're going to benefit from that. If anything, it's got harder at the moment to do other research just for practical reasons, and there's a big focus on COVID. I think, from my point of view, which is I sort of alluded to before, I do think there's a greater acceptance of doing stuff remotely, and that for rare conditions is going to be much more helpful. So we're having discussion with the UK drug regulator, not the European one anymore, but the UK drug regulator about about remote assessments and what sort of things are acceptable in terms of safety and efficacy assessments, and what we can do across videos and things, I think that conversation is easier now. So yeah, that's probably the main thing that I can think of. But yeah.
Hey, I'm sorry. I have to save some face here for a second. I wasn't capitalizing. So when you capitalize Vineland in Citizen, I immediately go to 3 pages in his record where the Vineland is noted, and I click on it, and I can see the exact page of his 6,000 pages of medical records where his Vineland results are mentioned or noted. So just offering it up, if you want to expand your cohort quickly and easily, we have almost 100 people who have this at their fingertips, and they'd all be happy to. And the IRB is written in such a way that you could easily get access to this data. Okay. Sorry.
Yeah.
Yeah, and I think speaking as a British parent in living in the US, I do see them doing a lot more of these standardized evaluations when you're going through the process of getting an autism diagnosis, and even just for entry into preschool or regular kindergarten, you need to go through the Vineland. So all parents should have these available to them of their children of a certain age. So we're working very hard to, talk to families about how to participate in trials, convince them of the benefit of participating in the studies, that we are a very, very small population, so every single person matters, every single child counts in terms of learning more about the disease. So we're trying very hard to recruit as many people as we can, and definitely would love to spread the word about the Neuro-GD study.
Thanks.
And others.
Yeah, I mean, and that's, I think, and we appreciate, these things take time. You guys are busy, but it is, as I say, if people can, it is, as you say, every person counts. If you're recruiting 40 people, every person is 2.5% of your total population, so it adds up very quickly what one person's data can do. So,
and this is why we're so gung-ho on Citizen, right? Because people are busy, parents are exhausted, and you say to a parent, what happened 2 years ago? I don't know what happened 2 weeks ago, number one. Number two, if it wasn't for Citizen, there's no way on God's green earth I would be able to find that report. So for longitudinal data, how is it, these patients, we can now go back, the Vineland was 2 years ago, 3 years ago, and look at how patient X is proceeding.
Yeah. And also, those are clinical appointments that you are going to anyway. It's not like you're traveling separately to come for a research study, so this taking advantage and leveraging the data that's already there is great.
Okay, so we did go over by 30 minutes. We all really appreciate your time, Dr. Stanfield. And if anyone has any follow-up questions, please do email us or get in contact with us. I'm sure we can forward those on to Dr. Stanfield's team.
Yeah, I realized right at the beginning, I normally stick my email up at the end, but I didn't. But you can find me online. I mean, it's just andrew.stanfield@ed.ac.uk, and I'm happy to hear from people. Those of you who do email me from time to time will know I'm not the best at getting back immediately, but I do usually get back. So, and if I don't, you can email me again. I don't mind repeat emailing. So yeah,
and to everyone who's still listening, I would advocate for participating in the Neuro-GD study. Aisling is a lovely person to speak to. I spent a good hour with her talking about your child, and then this packet of forms to fill out. So it's obviously not as fast as Citizen, but it's definitely worthwhile to take part in that. So get in contact with SRF, get in contact with Dr. Stanfield, and we can forward you the details of how to get involved in that study.
Thank you both. This is lovely.
Thank you.
Excellent. Thank you very much.
Thank you, and good evening.
Thanks now. Bye-bye.